Today we will discuss calorimetry in identifying partially folded states of proteins. We have earlier discussed the significance of understanding the protein folding problem. Since we do not know the protein folding code, therefore, it becomes very important to understand about the intermediates which occur during the protein folding. And as we discussed earlier, lot of efforts are being directed towards that, and slowly we are understanding more and more about this protein folding problem. And if we can understand the intermediates which occur during the protein folding pathway in a qualitative and quantitative manner, that is a contribution towards understanding protein folding problem. The protein folding is quite fast process, and therefore, studying the kinetic intermediates which occur during protein folding becomes relatively difficult. It will be very important if we can isolate such intermediate states under equilibrium conditions and then study these intermediates both qualitatively and quantitatively. We can get lot more information about these intermediate states. One such intermediate state which became very popular in late 80s and early 90s was molten globule state. And today we will specifically focus on the molten globule state or partially folded states. What are these states and how we can use the concepts of thermodynamics in identifying these states and getting qualitative and quantitative information about these states. Let us discuss this in details. Let us take a look at this slide. We are revisiting the protein folding problem. The protein folding problem is how this primary sequence folds into a unique three-dimensional conformation. This question mark is the protein folding problem. Why it folds into a unique three-dimensional conformation? And then we discussed that one of the most popular model which has been used in explaining the protein folding is energy landscape theory. And there I highlighted the molten globule state / partially folded states. This becomes more clearer in the next slide, that after the protein is synthesized from ribosomes and this primary sequence of amino acids, it has to adopt secondary structure and finally get the minimum energy three-dimensional conformation, which is the native state of the protein, and it goes through several intermediate states. Today we are going to discuss that if we can get qualitative and quantitative information about such intermediate states, then it is a contribution towards the protein folding problem. Now, as I mentioned, in late 1980s and early 1990s, a term molten globule state became very popular. As the name itself suggests, molten globule state. It is the molten form of the globular protein. Molten in the sense that it is not a rigid three-dimensional structure, but it can be in between the native and denatured state of a protein. Let us take a look at this figure. This figure represents a cartoon picture of the formation of molten globule state, and this was proposed by Tritson in 1995, where this part of the figure represents the native state and this part of the figure represents the molten globule state. And the difference between the two is some of the structural feature of the native state are retained, but some of the structural features of the native state are lost. So, this is a relatively molten form of the native state, and that is what is commented over here. The model of the molten globule state, according to this model, the molten globule preserves the mean overall structural features of the native protein, but differs from the native state mainly by looser packing and higher mobility of loops and ends of the protein chain. Before we actually start discussing the thermodynamics of the molten globule state, it will be very important to first understand what is molten globule state. Let us discuss a little bit more about this, and also what is the relevance of the molten globule state with the actual intermediate states which are observed in the protein folding pathways. Let us take a look at the comments. Different experiments over the years have proved that the hypothesis of intermediate state is indeed true. Several groups have been working from different angles in understanding the protein folding pathways and hence the protein folding intermediates. Lot of spectroscopic techniques have been applied to get qualitative information on the protein folding intermediates. However, along with the qualitative information, getting quantitative information becomes very, very important. Direct application of calorimetry can give quantitative information. We will discuss how that information can be achieved in today's lecture. Let us take a look at the next comment. Intermediates obtained under equilibrium conditions have similar structural and energetic properties as kinetic folding intermediates. This comment is based upon the literature information. Since the protein folding is a very fast process, if we can isolate the intermediate structures, intermediate states under equilibrium conditions, it becomes easy to study those. Literature suggests that it is possible to obtain intermediate structures under equilibrium conditions, and we will discuss a little bit more about that a bit later. Now, let us take a look at the next comment. Molten globule, partially folded states of proteins. Let us discuss what is a traditionally accepted definition of molten globule state. Several researchers defined molten globule state of a protein as a state in which the secondary structure is similar to that of the native protein or even strengthened, but the tertiary structure is poorly defined or tertiary structure is lost. So, therefore, the molten globule state a sort of became a specific title for a state in which the secondary structure is retained, but the tertiary structure is lost. Let me also make it very clear over here, since we are discussing about some specific research area today, it is not always true that a given terminology is accepted by different authors or different researchers. Molten globule state anyway is a state which is in between the native state and the denatured state. And as I said that several researchers defined the molten globule state as a state which specifically has no tertiary structure or poorly defined tertiary structure, but the secondary structure almost same as the native protein. Whereas some set of researchers believed that any intermediate state which is in between the native state and the fully denatured state is an intermediate state, and getting information about those intermediate states is anyway important, but they did not believe the concept of the molten globule state. So, this controversy one may find in literature, but nevertheless there are several reports which have demonstrated that such intermediate states do exist, in which the tertiary structure is lost or poorly defined, but secondary structure is intact. Now, let us take a look at the next comment. The A state of alpha lactalbumin, LA, I am using abbreviation of alpha lactalbumin, has become a paradigm for evaluating the properties of partially folded states. I will take several examples by using the protein alpha lactalbumin. Alpha lactalbumin is a milk protein. It has calcium in it. What happens is that if you remove calcium from alpha lactalbumin, it goes to an A state, it is called an A state, which is the molten globule state. That means, if you remove calcium from alpha lactalbumin, the tertiary structure becomes poorly defined, but the secondary structure remains intact. We will discuss more about this. Now, the question is that if we want to generate these partially folded states or so called molten globule state in laboratory, what should be the experimental conditions? What kind of additives we can use to generate these partially folded states? We know that urea can denature the protein at a certain concentration or above a certain concentration. Usually 6 molar or 8 molar urea is sufficient to denature a protein. Similarly, guanidinium hydrochloride is a protein denaturant. So, therefore, if we use an additive or a co-solvent which can partially destabilize the protein, partially denature the protein, should assist in pushing the native state to an intermediate state. And let us take a look at the slide, see what kind of additives can be used to generate partially folded state. One is pH. We have discussed pH at length. How pH can affect the conformational stability of a protein. Suppose if we keep on increasing the pH or decreasing the pH, both extremes of the pH can lead to denaturation of a protein. For example, if we keep on decreasing the pH, at very low pH, let us say at pH 1.5 or 1, the protein becomes very, very strongly protonated and there is a lot of positive charge. And due to the positive charge repulsion, the protein may unfold, it may undergo denaturation. And the same thing can be expected when the pH is very, very high. So, therefore, by altering the pH, some proteins may show a conformation which resembles a molten globule like conformation. Now, let us take a look at the next possibility. Urea. Urea is another molecule which affects the protein conformation. It is believed that urea can affect the protein conformation by altering the structure of water. Because urea can form hydrogen bonds with water, it will alter the structure of water to large extent. And alteration in structure of water can ultimately affect the protein conformation. Urea can also disrupt the protein structure by directly interfering in the intramolecular hydrogen bonding in protein. So, therefore, urea is another molecule which can push the native state of a protein to an intermediate structure. Now, let us take a look at the next one. Next is guanidinium hydrochloride or guanidinium chloride. Guanidinium chloride is another molecule which is known as a protein denaturant. Literature search suggests that there are several alcohols. For example, TFE 2,2,2-trifluoroethanol or 1,1,1,3,3,3 hexafluoroisopropanol, which is abbreviated as HFIP. These two proteins have been extensively used in literature to generate the molten globule like structure. This trifluoro group or hexafluoro group actually acts as a hydrophobic group, and what has been observed in literature that at certain concentration of TFE and HFIP, the tertiary structure of a protein is lost and the secondary structure of the selected proteins remain intact. And this is the conformation in which the tertiary structure is poorly defined or tertiary structure is lost, but secondary structure is intact, is termed as molten globule state. And let us again go back to the slide. The other alcohols which have been used to generate the molten globule like structure is 4-chlorobutanol and 2-chloroethanol. The next category can be salts. Let me discuss how salts can be used to generate the partially folded state or molten globule like state in a protein. I just discussed that if we keep on lowering the pH, at very low pH, extensively positive charge will be there on the protein because of strong protonation. And that excessive positive charge, due to positive charge repulsion, the protein may denature. Now, at very low pH, if we introduce large anions, these anions can neutralize the positive charge to certain extent and will induce refolding in the protein. And this refolding can induce a molten globule like conformation. And in other words, the refolding will lead to formation of certain intermediate conformations. That is how salt becomes important. And similarly, there can be other variety of experimental conditions which can help in generating the partially folded states of the protein. Now, the next question is, before we actually address the thermodynamic aspects, first we need to have characterization of these partially folded states. What kind of methods can be used for a qualitative characterization of these partially folded states? And if we are interested specifically in addressing the molten globule like conformation, in which the tertiary structure is poorly defined and secondary structure is intact, we should use a method which can directly give us information about the secondary structure and tertiary structure of a protein. And there are spectroscopic methods available. One such spectroscopy which directly gives information about the secondary structure and tertiary structure of a protein is circulardichroism spectroscopy. The principle of circulardichroism spectroscopy you will learn somewhere else, but let me very quickly tell you what the circulardichroism spectroscopy measures and how we can use it like a fingerprinting technique to get information about the secondary structure and tertiary structure of a protein. It not only gives information about the proteins, it can also be extended to other biological systems including DNA. Let us go to the slide. In circulardichroism spectroscopy, the sample, the chiral sample, the sample which have molecules having chirality, will absorb left circularly polarized light and right circularly polarized light to different extents. And it is the difference in the absorption which is measured in the circulardichroism spectroscopy. As we can see in this figure, let us say one of these absorption we take as left circularly polarized light and the other is right circularly polarized light, and the difference is measured in circulardichroism spectroscopy. The main features that we need to know are that CD measures the differences between absorption of left and right handed circularly polarized light. And let us keep in mind the next comment also, that after passing through the sample, left and right circularly polarized lights have different amplitudes, and the combination of the two unequal beams gives elliptically polarized light. I suggest you to go through the principle of circulardichroism spectroscopy if you would like to learn more in details how the elliptically polarized light is obtained. And hence the CD measures the ellipticity of the transmitted light, the light that remains, that is not absorbed, and pictorially it is represented in this figure. Now, this ellipticity as a function of wavelength can be correlated to the secondary structural and tertiary structural content of a protein. Let us quickly take a look at near UV-CD and far UV-CD features. Near UV-CD, which is in the range of 250 to 350 nanometer, is dominated by phenylalanine, tyrosine, tryptophan and disulfides. And the phenylalanine, tyrosine, tryptophan, disulfide basically define the tertiary structure of a protein. That means near UV CD signals will carry information about the tertiary structure of a protein, and the far UV CD, which is in the range 180 to 250 nanometer, in which amide group is most abundant, chromophore, CD chromophore in proteins, give rise to bands. For example, pi star transition will be observed at 190 nanometer and n pi star transition observed at 220 nanometer. But what is more important at this point is for understanding qualitatively the secondary and tertiary structure of a protein, the numbers presented in this table are important. For example, the different secondary structural elements, for example, alpha helix will show a negative band at 220 nanometer and a positive band at 192 nanometer. And alpha helix is also accompanied by a negative band at 208 nanometer. So, the bands at 208, 222 negative bands and a positive band at 192 nanometer will be characteristic of alpha helix. A negative band at 216 nanometer and a positive band at 192 nanometer are characteristic of beta sheet. Beta turn is accompanied by weak negative band in this range 220 to 230 nanometer and a strong negative band in 180 to 190 nanometer and a positive band at 205 nanometer. Similarly, a random coil will show up in negative band at 200 nanometer. So, these numbers can help us in qualitatively establishing the secondary structure and tertiary structure of a protein, and the changes in ellipticity as a function of temperature or as a function of the concentration of additive can tell us that how relatively the extent of secondary structure or tertiary structure changes when the environment is changed. Let us take a look at the next slide. Now, these are the far UV CD characteristics for alpha helix. The red one, you see two bands, two negative bands. These characteristic, this kind of spectra is characteristic of alpha helix. Beta sheet is showing one negative band and one positive band here, and the random coil is showing a negative band. The numbers were given in the table that I showed in the previous slide. Now, let us take a look at the next slide. We will discuss about the molten globule state formation and its thermodynamic characterization in the next lecture, but alpha lactalbumin at pH 7 without the removal of calcium ion shows a molten globule like state in presence of 1,1,1,3,3,3 hexafluoroisopropanol. And we have just discussed that how circular dichroism spectroscopy can be qualitatively used in establishing the molten globule like state. If we look at the upper panels, these are the far UVCD and near UVCD spectra of alpha lactalbumin in presence of different concentrations of hexafluoroisopropanol. For the time being, if we just concentrate on the blue line, it shows two negative bands at about 205 and 222 nanometer, characteristic of alpha helix. Very strong secondary structure, just similar to that of the native one. Native one is shown over here, 0 molar. That means the secondary structure is relatively stronger than that in the native structure, but look at the blue line here, the electricity is almost 0 in presence of 0.5 molar hexafluoroisopropanol, tertiary structure is lost. So, in presence of 0.5 molar hexafluoroisopropanol, secondary structure is strengthened, tertiary structure is lost, and therefore, this concentration induces molten globule structure, molten globule conformation in the protein. The lower panel is fluorescence characterization, and that we will discuss a little later. But the circular dichroism spectroscopy is directly telling us that 0.5 molar hexafluoroisopropanol induces a molten globule state in the protein. So, what we have discussed in this lecture is that what is the significance of studying the partially folded states of a protein, what is a molten globule state, how to experimentally obtain a molten globule state under equilibrium condition, and how to characterize a molten globule state in a qualitative manner. That characterization we have discussed only by using one technique so far now, that is the circular dichroism spectroscope. We will try to understand more about quantitative aspects and associate the thermodynamic parameters along with it, and discuss how to obtain those thermodynamic parameters, how to get quantitative information about the molten globule state and such partially folded states in the next lecture. Thank you very much.